Fertility is the ability of a woman to conceive a child and is dependent on ovulation, which is the release of an ovum or a mature egg from the ovary. Ovaries contain thousands of follicles, each containing an immature egg or oocyte. Follicles release hormones into the bloodstream and nourish the developing oocyte until it becomes a mature ovum. This ovum is then released from the follicle into the fallopian tube. Once in the fallopian tube, the ovum can be fertilized by a sperm. If fertilized, this fertilized egg will travel to the uterus to implant, beginning pregnancy. Although fertility is dependent on ovulation, many factors can prevent or delay ovulation. Many women are left infertile after enduring cancer treatments. Chemotherapy is a drug treatment that is intended to attack and prevent the growth of cancer cells by damaging the genes inside a dividing cell's nucleus. While it is an effective cancer treatment, chemotherapy also attacks growing ovarian follicles, leading to the destruction of oocytes, follicle depletion, and ovarian cortical fibrosis and blood vessel damage. In response to this infertility issue, researchers Teresa Woodruff and Ramil Shah have constructed bioprosthetic functional ovaries for mice using a 3D printer. The printer creates a scaffold, or skeleton of the ovary, using the structure of a healthy, normal ovary as a model. Once the scaffold is implanted, it supports the survival of oocytes, which can then be matured to ovums and ovulation can return. The printer uses gelatin made from broken down collagen as its ink, or material to build the scaffold. Gelatin prevents the artificial ovary from collapsing while also allowing it to naturally interact with other body tissues. A 3D printer creates these ovary scaffolds by overlapping various patterns of gelatin filaments in a way that the researchers say is similar to building with Lincoln logs. After printing, the scaffolds are then injected with follicles. Once implanted, blood vessels are able to enter and grow through the ovarian scaffold, allowing for vascularization that is crucial to delivering nutrients to the follicles and circulating follicle-produced hormones through the bloodstream. After removing the ovaries of mice and implanting these 3D printed ovarian scaffolds, these previously infertile mice were able to successfully ovulate, conceive, and give birth to healthy pups. Because mice can function as model systems for humans, the results of this experiment are incredibly applicable to humans. A model system, also referred to as a model organism, is a non-human species that is studied in order to understand and make discoveries about biological phenomena. Model systems are useful because they are easy to maintain and modify in a laboratory setting and can inform scientists on human biology at times when it would be unethical to study humans directly. Due to model systems, experiments which can better the lives of humans in the future can be completed without subjecting humans to much risk. Through further experimentation, scientists hope to eventually restore full fertility and hormone production to human patients that have become infertile due to cancer treatments.